Okay, so my name is Carter Pete. This is my DNA replication project. This is my strand of DNA with matching nitrogen base pairs. I don't know if you can see if they're matching, and it's twisted into a double helix. And the first thing that happens in DNA replication, well, not the first thing, but the first thing that we're covering is it goes to the topoisomerase, which will then straighten it out from the double helix into a more easily dividable straight line. And then the helicase, which is neatly labeled there, will then split the DNA into two. It doesn't split it all at once. It splits it section by section, like this. And so now you've got two strands. The leading strand, which has the 5' that way and the 3' this way, can just put the primer on the end, because DNA polymerase can't build off nothing. It has to have a primer there from RNA, which matches up with U's instead of A's. And then so the primase matches that up, and then DNA polymerase can just go along and match up the nucleotides. So right here it would be GGC. So and the DNA polymerase only builds 5' to 3'. And so as this strand comes out, it can just keep on building off the primer. So that's why it's called a leading strand, because it can just keep on building. This strand, on the other hand, has a much more difficult time, the lagging strand. This is the side that comes out 3'. So it starts, since the DNA polymerase can only build 5' to 3', it has to start right here, but it needs a primer too. So it takes the primer right there, and then it builds, it takes the primer right there, and then it builds off of that, except it reaches the end. So what needs to happen is it needs to wait for the helicase to cut more DNA. So you've got a strand right there with a primer and a bunch of RNA and several nitrogen bases. And then it can go back. Primase will lay down another primer right there, go back to where it left off, and then it will add in the remaining bases right there. And that's why you have Okazaki fragments of the lagging strand, because the lagging strand can't build continuously. It has to lay down a primer, build to the end, and then go back, lay down a primer, and then build to where the last primer was. And also, in order to keep the strands from coming together, you've got single-stranded binding proteins that attach to the outside of each of the DNA strands. They keep them separated. Okay, so the ligase also, what it does is after everything's complete, it goes in and it seals everything off to finish the job, removes the RNA primers, and puts down the correct nitrogen bases, but the DNA nitrogen bases this time. So it basically reconnects all the Okazaki fragments and stops them. It makes it all DNA, and then it seals it all up, and you're good to go with your new DNA.